Okay, so hello everybody. Welcome to this our room one of our short parallel talks, our trainee talk. So again, the idea here is that our speakers are going to speak for approximately 5 minutes. Think about this sort of like, what your poster spiel would be like if you were standing by your poster at SFN or CNS or something like that. And then we're going to leave about 5 minutes after that for interactive Q&A. Again, just like if you were at a poster session, afterwards, when you hear spiel, you would sort of, talk back to the presenter. So we're trying to, get that experience in that environment as much as possible in this interactive setting. So to start off today, we have two members of the Campbell Lab, Sue Ann Campbell, who presented this morning. Maria Chugnova is up first, and she's going to talk about inhibition by calcium as a synchronizing mechanism in gonadotropin-releasing hormone neurons. So go ahead, Marina.
Thank you very much. Thank you to organizers, and thank you, everybody. Looks like we have pretty nice shindig. The object of our study is GnRH neuron, and I will call it neuron next. This is the portrait of my beauty, and it has so unique and unparalleled morphology, structure, so I wouldn't even dare in 5 minutes even to list all of those. I will just say that actually the projections of this neuron works both as axons and dendrites, and they project to medium eminence where they secrete that hormone, GnRH. And only the synchronized surge of this hormone triggers the whole reproductive system. The next step, this important information for us now, is the next endocrine cells, which secrete in their order LH and FSH. I will just sort of shortcut to save some time. You can read what it means. Problem number one, with very complex and actually very vast electrical machinery, this neuron does not produce that surge of hormone, even if there is full synchronization of electrical signal, which is very surprising, but this is experimental fact, which is the law for us. So let's consider a single neuron of this type, which is by the previous lectures is level 0, and every neuron is actually self-sustained oscillator, and it includes electrical activity inside and outside calcium, and these hormone receptors which activate 3 subunits of G protein. 2 of them activate the calcium release, and 1 of them is inhibitor. As a result, we used modified and tuned a little bit the model created by Dr. Kadri, Dr. Lee, and by that modification, we obtained the oscillation of a single neuron, which is pretty close to the experimental data. And as a synchronized model, we suggested the diffusion of this agent, of this hormone, between different neurons of this type, suggesting that this diffusion can synchronize the oscillation of that magical surge, which triggers the whole reproductive system. So as usual for diffusion, we have PDEs, and we included into previous model the coupling term, and as a result, synchronization looks very nicely. This is synchronization of oscillation of one neuron, and this is oscillation extracellular, as you see the surge. And if we'd have more time, I would show you that actually, again, this is pretty good according to experimental data. However, all these models are based on the assumption that those neurons are homogeneous. That assumption was good until actually the studies in vivo started, and these neurons are not abundant very much, a maximum 2,000, and location is extremely difficult to reach. So in vivo, experimental in vivo started only in 2013. The more experimental data collected, the more it becomes clear that actually those neurons are heterogeneous. So what to do? Here comes another fact. Again, recently discovered Kisspeptin neurons produce Kisspeptin, which activates electrical activity, and this activation actually generates in many ways the increase of calcium transients in that neuron. So simply speaking, the concentration of calcium increases many times. It's not only the theory, it's also confirmed by experiment. Indeed, increase of calcium happening in a very strange way, in a way as it was described in this article. It looks like the whole volume of neuron increases its calcium concentration, not just at one level. So here comes our conjecture that we're working on at this moment. So we conjecture that this sharply increased and sustained level of calcium ions throughout the whole volume of GnRH neurons inhibits the secretion of that hormone, and by that resetting that secretion produces that synchronized hormone surge. What do we mean? These are the oscillations in heterogeneous neurons. You see, they are sort of different in shape, in shift, and in amplitude, but as a result, they can't produce the surge. But if this increase of calcium ions happens throughout the whole neuron, then they actually stop oscillating altogether, and then they start producing that surge with exactly the period we're expecting them to do. So that's what we work at the same moment. And actually, last week, suddenly we received some encouragement in this article. This is review in Frontiers of Endocrinology. It was published in 2013, and it's not exactly about this neuron, but this is about the neurons that produce the next step of surge of LH and FSH. Those hormones also produced in many ways in similar manner, but after a lot of review, a lot of research, a lot of experimental comparison, the authors of that article concluded that actually to produce those surges in their order, it's supposed to happen, this sudden and sharp increase in calcium ions in the whole entire cell. So it's really close to what we are thinking about. And at this moment, we are looking for the mechanisms and the ways to describe those with the equations. And it seems like we found some by this time. Thank you very much.
Awesome. Thank you, Marina. So we only have a few minutes for questions here. I guess, so my first question would be, so you've created this very detailed single neuron model, and you're talking about synchronization. You're
Yes.
talking about synchronization of the neuron's activity with extracellular activity, correct? It's not sort of neuron spiking synchronization?
We were planning to do that, but Dr. Snade and Dr. Leon already beat us to that. They created fantastic model, though we are planning to still improve it. And we are not focused very much. This is Chen and Snade. They published an article in 2015, and it's actually based on 2000 Le Bohr model of electrical activity of that neuron, which is also, it's just a masterpiece. So these are good models, and we are okay for now with it. And this is not the center of our sort of attention right now, just because, as I was telling in the beginning, in problem number 1, experiments, very good experiments, they do it in New Zealand. They proved already that just synchronization of electrical signal do not create that surge. So that's why we're okay with that model for now.
Awesome. So let's all virtually thank Marina once again. I'm now going to sort of boot you from the screen, Marina, so I can invite up our next speaker. So Malia, I'm going to now invite you onto the screen. Hello. So go ahead and share your screen with your presentation. Fantastic. So okay, so now you probably can't see us, but we see your PowerPoint. Yeah, so I'll sort of be the voice in your ear moving forward. So for our second talk today, we're happy to have Malia Ahmed, who again works with Dr. Sue-Ann Campbell, and she's going to talk about how a mean field model for a network of 2-compartment integrate and fire neurons.
Thank you so much. Hello, everyone. So my name is Malia Ahmed, and I'm a PhD student at the University of Waterloo. And I am, as Scott mentioned, I'm working under the supervision of Dr. Sue-Ann Campbell. And so today I'll be presenting to you a mean field model for a network of 2-compartment integrate and fire neurons. So the derivation involves using a population density approach together with a moment closure method and a separation of timescales, which is based on a steady state approximation. So typically population density methods involve large network of point neurons, which lack any kind of spatial structure. And so we considered a 2-compartment soma and dendrite model for the individual neurons. And what I've included down here are the set of equations that for a network of n such 2-compartment neurons. So the state variables we have are the soma voltage, the adaptation variable for the soma, and the dendrite voltage. And I've also highlighted the synaptic coupling term because it's important in its relation to a population firing rate. So step 1, we use the population density method, which refers to the limit as n, which is the number of neurons, goes to infinity. And so instead of simulating and keeping track of all the state variables of the neurons individually, the idea is to group similar neurons into populations and simulate their distribution over the state space. So the time evolution of the probability density function follows a conservation equation. And in the large network regime, or in the large n regime rather, the synaptic coupling term needs to be replaced by the population firing rate, which is down here. So we've converted our full network into this using the population density method. And in step 2, we reduce the dimension of our system by applying the moment closure method. And there are 3 key assumptions here. So the first one is that the first conditional moment, which is the mean of W given Vs and Vd, and the Vd given Vs is equal to their corresponding unconditioned moments. Then we rewrite everything in terms of the marginal densities And in the simplification, at one point, we need to make this second assumption that the mean adaptation is much larger than the size of the individual jumps. And the third assumption is really a working assumption in order to simplify and work with this nonlinearity that shows up here, and in order to pull out this S outside of the integral. So the assumption is that S of t is independent of VD and W. And in our step 3, we apply a quasi-steady state approximation. And it's based on the assumption that both the density and VD reach their steady state values faster than S and the mean adaptation. So we've got these two equations that we've set to 0 now using this steady state approximation. And it really is this third step that takes us towards a mean field model, which looks like the following. So we've really reduced our dimensionality as well as the number of equations that need to be solved. So we've got a single equation now that representing the evolution of the mean adaptation, and we've still got our synapse equation. Both of these are dependent on the population firing rate, and this itself has some conditions, such as we need the denominator to be non-zero and so forth, that I haven't included here. We've also got this algebraic equation now. And then with this, we ran some simulations using Euler integration and 1,000 neurons. And what you're seeing here is in orange, it's the true network average using the full model. And in black is the mean adaptation. So this is for the adaptation variable. In black, we're seeing the mean adaptation using the mean field model. And what you can really see is that it does indeed give a good fit. And similarly for the synaptic conductance, in blue, we're seeing the synaptic conductance for a single randomly chosen neuron from the full network. And in black is the synaptic conductance that we are simulating using the mean field model. Once again, this approximation does indeed give us a really good fit of the full model. Now, this work can be considered as the first few steps, I'd say, in working with large networks that include some basic neuronal morphology. That was one of our key things, that we wanted to include the soma and the dendrite, which at the same time can be reasonable to work with computationally. So the methods that I've shown here are not limited to a specific choice of neuron model. It can be generalized, definitely. And my hope is that we can use this towards coupling different populations, not just a single population of excitatory neurons as we have here, and arriving at working on large thalamocortical networks. And with that, I thank you. And here are some of my references.
Awesome. Thank you, Malia. So I'll start off the questions, and again, please post your questions in the chat or in the ask a question box. So I guess my first question to you would be, there's obviously some parallels here between, other population models, Wilson-Cowan networks, things like that. But obviously you're taking this, spatial component of neurons into account, which is very important. What did you find was sort of the biggest differentiating factor between your population model, where you take that into account, as opposed to some of the existing population models that primarily think about things as point neurons?
I think the first thing is, because now we've increased the dimensionality of our state space, that posed some challenges in that we had to make some extra assumptions. Like, for example, I think I mentioned here, this third assumption really was just a way of simplifying our work further. And so I think working with not including the dendrites, for example, or starting off with a two-dimensional model is, as for example, some of the other models have done, is I guess, it doesn't require as many assumptions as including more variables does, and increasing the dimension of our state space. I think that was the biggest thing that I found different.
So do you think that, so a biologist may look at this and sort of ask you, okay, well, are these assumptions sort of, what's the underlying sort of biological justification for these assumptions? That's the hard question we always get. So So what would your response be to someone who asks something like that?
I think that that's definitely something Suhan and I have discussed as well, that it's these are more so assumptions that are mathematical in nature, and certainly mathematical in nature, and it's more of okay, we're making an assumption that this is how our simplifications should be, and whether or not we are getting the right result. For example, in our case, we're looking at whether or not having this average approximation is indeed a good approximation to the full model. So I think, modeling in general is really your making really big or approximations to the full problem. So I think the field of modeling itself is such that I think the mathematical assumptions that we're making are reasonable.
Yeah, no, that's. I asked you the tough question there, so that was a very good answer. That's something that for any model is a tough thing to answer, and I think depending on the questions you're trying to answer, the answer being, well, we have to do that for the math to work, is completely reasonable, just as long as you follow that through the entire work, which it looks like you've done. Right. So any other questions from the peanut gallery? Yes, we have a question. So Alexandra asked, your SFT showed some variation relative to your mean field model. Do you think that's concerning? Would you like to sort of better fit that, or is that something that's a necessary side effect?
Is this the SFT that Alexandra is talking?
I'll see if she pipes up in the chat. I believe so.
Is it a concern? I would say not. The other thing that I would have wanted to plot, so these are simulations that we've done very recently. The other thing that I would have wanted to check is to plot also the population firing rate and see how that varies. But I'm not too surprised by this blue curve, because again, we're talking about this is coming from the full network model equations, and so we've got interactions between all the other neurons, whereas in the mean field we're averaging that out. So I'm not too surprised by how it's not tracking it, if that's what the question is referring to.
Yeah, no, I think that makes perfect sense to me. Awesome. So thank you very much, Malia. I'm now going to unceremoniously kick you off screen. And so I'm now going to invite up Alexandra, who is our next speaker. Hello, Alexandra.
Hello, Scott. Here are my slides. You guys can see them?
Yes. So go ahead and share your slides.
Okay. Let's see. Let me know if you can see them.
Not getting anything yet. So did you do the...
I did the share screen. Yes. All right. One more time. Now it should work.
There we go.
Okay, perfect.
Okay, so we've got your slides up. So I'm happy to introduce Alexandra. Should I even try?
It's okay, I'll do it for you.
I was all revving myself up to try, and now I'm probably just gonna embarrass myself. Anyway, Alexandra is one of my colleagues in the Skinner Lab, and she is going to talk a little bit about sort of continuing what Francis talked about this morning about these in this interaction between detailed and more microcircuit level models of theta.
Okay, so thank you, Scott. Thanks to all the organizers for this opportunity. So as Scott mentioned, my name is Alexandra Hedzikaliniu, and I'm a PhD candidate with Francis Skinner at the KCN Hub at the Krembil Research Center. So today I'm going to talk about how state-of-the-art biophysical models of the C1 region of the hippocampus reveal how theta rhythms are generated and how their frequencies are controlled. To answer this question, we use a state-of-the-art biophysical model developed originally in the Ivan Solsa's lab in Stanford by Bezier and colleagues in 2016. It is a model that contains 8 different inhibitory cell types. They're all specially extended, connected with pyramidal cells, based on empirically derived connectivity from experimental data. So this is a biologically very detailed model. And it requires a lot of computational resources to do any of those simulations, even for those who have access to supercomputers. Thus, to understand this model, we had to reduce it. We thus isolate a segment and 10% of its volume, shown here, and we found that the segment model can also produce theta activities. The question is how? We have some open questions. Even though our system produces theta rhythms, we don't understand the dynamic behavior. We don't know, for example, how the frequency and the power depend on the underlying network parameters. We also don't know how the theta rhythms even initiated. So here's a schematic of the underlying network connectivity map. We focus on synaptic conductances. We know from comparison with the experimental context that if a hippocampus is placed in an excitable medium, that is an intact ex vivo hippocampus preparation, it can self-organize into a theta-generating network, likely because it's activated by remaining afferents from entorhinal cortex and the CA3. So we also know from a comparison with a minimal model that Dr. Francis Skinner talked about this morning, that there was a huge discrepancy between the two models on the level of the excitatory conductances delivered onto the pyramidal cells. Those are the recurrent pyramidal conductances and the entorhinal cortex and CA3 to pyramidal conductances. Thus, we focus our investigation around those conductances and also around the level of stimulation, which represents the level of activation of the entorhinal cortex and the CA3, and that's delivered to the entire network. So we find that the theta power increases monotonically with both EC-CA3 and pyramidal to pyramidal conductances. And you can see that here. For the large values of those conductances, there's a sudden decrease in this pattern. And that's because our networks, as a matter of fact, shift from theta generating to delta generating network. So if we were looking at 1 to 12 hertz range, for example, we would have seen only a monotonic increase in the power here. We can see the same disturbance for the theta frequency. If we take a look at the stimulation, we find that it is almost anti-correlated with the normalized theta power. And we can understand that if we take a look at the inner workings of the network. So here we're going to look at 2 cases, A and B. In case A, the synaptic conductance from the entorhinal cortex and C3 to the pyramidal cells is low. For that reason, a strong concurrent activation of the external afferents is required to induce a certain amount of activation in the pyramidal cell population. Now, because the stimulation is large, there is a strong concurrent activation of most of the interneurons in the network. In case B, on the other hand, because the EC-CA3 to pyramidal is large, less stimulation is required to induce the same amount of activation in the pyramidal cells. For that reason, the concurrent activation of most of the interneurons in the network is low. By the way, this pattern is not true for the CCK cells. We're an exception. This tells us the following. We can induce the same amount of activation in the pyramidal cells, leading to networks of very similar theta power through different cellular pathways, which allows us to hypothesize the very reasonable thing that what really seems to be important here is the net amount of input to the pyramidal cells. So to examine this hypothesis, we analyze the 50 networks that underpin our heat maps here. We take a sample of 10 cells per network, and we look at the average across cells of the mean input current to each one of those pyramidal cells. And we plot the theta frequency of the network as a function of that net current. We found that there is a very strong correlation between the net input current to the pyramidal cells and the resulting theta frequency, which allows us to say that really we can predict the frequency of the resulting network by looking at the net current delivered to the pyramidal cells. It's also worth mentioning here that because this is only a segment, a small fraction of the hippocampus, the interneurons have not yet had the chance, at least most of them, to organize into coherently firing populations. And as a result, the input to the pyramidal cells is very noisy. Despite that, if the amount of the input is appropriate, the pyramidal cells can self-organize into theta bursting populations, which allows us to say that it seems that the theta rhythm is initiated by those pyramidal cells. So in conclusion, we expose two cellular pathways of pyramidal cell activation, resulting in networks of equal theta power. We show that theta frequency is correlated with the net input delivered to the pyramidal cells. We found that the theta rhythm generation is not specifically due to phasic drives from inhibitory cells, at least in its initiation. It's really the net amount of input that can allow the pyramidal cells in the end to initiate the theta rhythm. Now, of course, as the network becomes larger and the inhibitory cells organize into coherently firing populations, they also contribute to stabilization and the control and the robustness of the theta rhythm. I would like to thank my supervisor for her support over the years, my colleagues and collaborators, especially Dr. Ivan Rykov in Stanford University, and my supervisory committee members. Thank you very much for your attention.
Awesome. Thank you, Alexandra. So I'll kick things off again while we already have a question in. Fantastic. So the first question for you here is this intrinsic theta oscillation? So there wasn't a mention of the medial septum. So is this a model of intrinsic theta oscillations?
That's right. I think I can also see the questions. Yes. So Here we're talking about the intrinsic theta oscillation. This is this intact hippocampus preparation. The experimental equivalent is this intact hippocampus preparation, which if it's isolated from the brain and it's placed in an excitable medium, it can on its own generate theta rhythms. And this speaks to the very initiation of those theta activities, which of course then are enhanced in vivo scenarios and as medial septum inputs also arrive, et cetera, et cetera.
Awesome. So while we wait for a couple more questions, my question here is, I'm always fascinated by the diversity of interneurons that you have in the large scale model. And I think that's one of the most interesting translations from the minimal model to the large scale model is taking into account that vast diversity of interneurons. So what do you think, what does that tell you about the sort of necessity for this heterogeneity, the necessity for these wide varieties of populations of interneurons that all have their own properties, connectivities, et cetera? What role does that play in theta?
Right. So this is my next paper. This is actually a problem that I recently kind of solved for my PhD. It was not meant to be presented in this talk. But really what I can say is that we have cells that are mainly driven by the pyramidal cells, and then we have cells that are mainly driven by external noisy sources. And that creates from starters, that creates two categories of interneurons. We have interneurons that really map the pyramidal cell activity, and we have interneurons that don't map this activity. And also within the interneurons that don't map this activity, we have interneurons that organize into coherently firing populations and interneurons that fire very noisy. So as a result, there is a circuitry that is being generated in which certain interneurons are fired because of the pyramidal cells, and then those interneurons control other gamma firing interneurons. And in turn, those sort of terminate the theta rhythm. But I can't say more about that right now because it's a sort of an ongoing finishing project. But we'll say that, yeah, you're going to find out from me really soon on that.
Awesome. I'm looking forward to it. Yeah. So we have one last question. So this is from Omera. So which intrinsic properties in the pyramidal cells make them or allow them to sort of initiate the theta oscillation?
So that's from Homera?
Yeah.
Yes. So as Homera knows, I've done some simulations to look at certain conductances, and I've looked at IH, for example, and I know that if we remove IH, then the oscillation becomes faster. And that is something that has to do with the intrinsic properties of the pyramidal cells. Like obviously, the intrinsic properties of the pyramidal cells are the ones that tune the pyramidal cell into a theta bursting population. So this is really the main take-home message from this presentation, is that the pyramidal cells define the frequency. And then that frequency is stabilized by interneurons with various mechanisms. But if you take away the IH, for example, then the rhythm becomes faster immediately.
Awesome. So thank you again, Alexandra. I will, again, unceremoniously now boot you from the screen so that I can invite up our final speaker. So let me find Ashraya here and invite her on screen. Hello.
Hi. Let me share my screen. Can everybody see it?
Yep. So we have your slides up now.
Yeah.
So yeah, for our last talk of this session, I'd like to introduce Ashraya Samba Shiva. Hopefully, I didn't
That's
butcher
fine.
that. So she is actually joining us from overseas in the UK. So thank you for staying up late. And she's going to talk about a population model of oscillatory dynamics in hippocampal CA1 and CA3.
Thank you, and thanks for organizing this. I'm Ashraya. I'm a part of the Graham Lab in University of Stirling. I'm based in Oslo, though. This is going to sound like an extended talk from what Alexandra actually presented, because I'm going to be focusing my talk on not the intrinsic oscillations, but the external, like from where hippocampus gets its theta oscillations and theta coupled gamma oscillations also. So go into the presentation mode here. Yeah. So I'm not going to go into a very detailed explanation of what the reasons of the hippocampus and stuff, but here I need to specify a few important pathways and the external inputs that the CA1 and CA3 region gets, which are important for my model here. So I'm going to be focusing on the entorhinal cortex input that comes to CA3 via the dentate gyrus and then to CA1, and the input it gets from the septum, and also the CA3 to CA1 input, and the recently discovered Bragg projection cells in CA1 to CA3 also. So these are certain important parameters that define my model when it comes to generating theta and theta coupled gamma oscillations. So in this, taking from the previous figure, the biological one, we scrapped down a lot of many other interneurons and stuff, but we have a very basic model of a microcircuit of CA1 and CA3. We have done simulations of CA1 and CA3 individually, and then we have integrated both of them. And this is the integrated model. And this is derived from actually the Denham and Boriscube model of the septohippocampal pacemaker circuit. So I'll try to explain it here. So the actual septum pacemaker circuit actually defines the generation and propagation of theta oscillations from septum to CA1 pyramidal cells. So that is coming from the septum to the other interneurons, which they do not have any other. It's only all interneurons gathered in one variable, so it's other interneurons. And then it comes to the pyramidal cells, which is propagated back to the calbindin containing hippocampal cells, which is given back to septum. So this circuit is pretty critical in CA1 to generate theta oscillations, which are propagated from septum. But we have also considered a couple of more bistratified and basket cells, and we've taken these projections from literature. We kind of extended the same for CA3 as well, and we found that it is possible to have the septal pacemaker circuit for CA3 also to generate theta oscillations propagated from the septum. Of course, without the septum also, we could generate theta and theta coupled gamma oscillations both in CA1 and in CA3. But since we are interested in septum propagated, we have the same pacemaker circuit in the CA3 also. And we have the external inputs from the entorhinal cortex to CA3 to CA1 and CA3, and the dentate gyrus input coming to CA3 pyramidal cells. Oops. But there are certain differences that I'd like to point out in these microcircuits between CA1 and CA3, two main differences, which is the basket cells and the bistratified cells in CA1, they get a bidirectional input here, for which there is evidence in literature. And also the septum input is propagated to bistratified cells here. But we do not see a bidirectional input from basket to bistratified cells in CA3, and there is no septal input to bistratified cells in CA3 as well. So this is the septum pacemaker circuit from the Denham and Boris-Duke model, and these are the original equations which they derived from Wilson and Cohen model. So it's a population model. So with this, we extended the same to include many other interneurons as well, mainly which we are interested, stratified in basket, and also the recently discovered back projection cells, which gives feedback to CA3. So we know from the papers that it projects to CA3 pyramidal cells and interneurons, but we do not know which one for sure. So we have projections to all the other interneurons that we have considered here. So the results. With the individual models, we could, this is the result of the integrated model, but I'd like to explain a bit about the individual models as well. In the individual models of CA1 and CA3, and with the integrated model, we see the exact same oscillations, theta oscillations as given here. So there was no difference in the phases or anything, but if we have to find differences between CA1 and CA3, it's the same with integrated and in the individual models, that we see this phase shift here in CA1, which also aligns with literature and which is not present in CA3. So in CA1, we see a phase shift for basket cells, which is not out of sync with pyramidal cells, and this can be eliminated if the basket cells gets more excitatory input from pyramidal cells, either from CA1 or from CA3. So this is true even in the integrated model. Like if the basket cells in CA1 gets more excitation, then this phase shift disappears, and it aligns with pyramidal cells and bistratified cells, just like in CA3. But if we try to induce even more excitation of basket cells, then that's when gamma coupling appears in CA1. And that's true in the integrated model and in the individual model. But this gamma coupling that will be there in both pyramidal and basket cells due to higher excitation of basket cells is limited only to CA1, and it is not propagated to CA3. But in CA3, when we try to inhibit the excitation of pyramidal cells, we get theta oscillations. So it was fairly easier for us to get theta oscillations in CA1, but it was quite a challenge to get theta oscillations in CA3. And when I tried to inhibit pyramidal cells, it was then that the frequency reduced, and we got theta oscillations in CA3 as well. And then when we try to induce excitation to basket cells, that's when we got theta coupled gamma oscillations here. And with this theta coupled gamma oscillations was not confined to CA3 alone, but obviously in the integrated model, we could see that even when this was in theta, when we had configured this for theta oscillations, when we had configured CA3 for gamma coupling, this kind of propagated to CA1 as well, and we could see gamma coupling in CA1 also. So we could safely say that the theta coupled gamma oscillations, or the gamma coupling here, was propagated from CA3 to CA1. So this is how it looks, the CA1 and CA3 in theta coupled gamma oscillations mode. And this is also from the integrated model. And in the individual model, when we obtained theta coupled gamma oscillations, we actually had a phase shift here with bistratified cells. So the bistratified cells in CA1 was out of sync with pyramidal and basket cells. But here in the integrated model, we do not see any such phase shifts. And that could be because the CA3 input that we are giving in the integrated model to CA1 is not an external constant like in the individual model. So it varies according to how this model varies as well. So it could be because of that. Now it's kind of getting the actual CA3 input instead of a constant input, and so we do not see any phase shift, and it's nicely aligned. When we increase the excitation when we decrease the excitation of bistratified cells, actually we also see gamma coupling in bistratified cells also. So that was another observation that we could arrive at. So other than that, one nice observation with the back projection cells was that when we try to vary the weights of the back projection cells in CA1, we could find that it was affecting CA1 back because of, obviously it will affect CA3 because of the projections to CA3, but because of the CA3 to CA1 projections, we could see noticeable differences in CA1 also. So it was important to have a proper weights for back projection cells, proper excitation of back projection cells in CA1 to have this kind of a nice theta gamma coupling to be maintained, or even theta oscillations to be maintained. When it comes to external inputs, we could see different kind of oscillatory states in the pyramidal cells in both CA1 and in CA3 by varying the external input values from steady state to theta oscillations to theta gamma coupling, and then to beyond 13 Hertz oscillations, and then it goes to even fast ripples beyond 100 Hertz oscillations also. So it depends on what kind of input we give for the external inputs, the entorhinal cortex or the septum or the dentate gyrus to get proper oscillations. So when wrong or higher or extreme inputs are given to these external inputs, we do not see a nice theta gamma coupling kind here, and that could possibly be also attributed to many mental disorders also. So there are a few limitations, though. We haven't studied a lot of other interneurons, inhibitory interneurons in this model. We can try to incorporate them also and see their effects in the theta gamma coupling here. And one of the actual limitations here is that with the population model, we cannot study or we cannot store and recall patterns. So no memory operation study can be done with this. So we are applying to take another approach to do that, using the cellular automata model for that, so that we can study both oscillations and to store and recall patterns here. So that's my talk. So any questions, I'd be happy to answer now.
Awesome. Thank you, Ashraya. So we have time for maybe one or two quick questions here. So there's already one here which asks, was the inhibition of pyramidal cells done by modifying a particular parameter in your model?
Yes. So all of these, the kind of oscillations that I want to get is based on changing the weights of each of the projections that I showed in the microcircuit. So yes, that's why that's why I said, like, when I tried to inhibit pyramidal cells in CA3, that I could get proper theta oscillations. And when I tried to excite more basket cells in CA1 and in CA3, either from pyramidal cells or from, either from pyramidal cells in CA1 or in CA3, I could get gamma coupling there in theta. So yes, it is based on modifying certain weight parameters. So it was obviously a lot of playing around with many of the parameters, but I had to find the exact parameter that affects the whole model and not just saying that all of them vary. So that's not right. So yes.
Awesome. So I guess really quickly, I'm just sort of curious because this is obviously a very detailed microcircuit, from a computational perspective, how long do these simulations take to run? How computationally intensive are they?
These are population models. They are not single neuron models, so it's fairly fast.
Okay,
but,
so
if it was a single neuron model, then that's when we cannot run computations for more than maybe 10 or 20 neurons at a time. And that's the reason it's really expensive computational wise. And that's one of the reasons why we are opting for a discrete kind of model now with the cellular automata, where we can also study with more number of neurons, which is like thousands. and also study storing of patterns, so that I wanted to study how memory actually works with respect to oscillations. Like here it converges. If I try to, I've also tried doing memory storing and recalling memory patterns with this, but then it just converges. No oscillations. So that's not what I want. So because the brain doesn't really converge when I try to recall something, right? So, so yeah.
Okay. Awesome. Well, thank you, Ashraya. Thank you to all of our speakers in this session. So we now, I'll sort of politely nudge you. We have a sort of concluding remarks session, just sort of 5-10 minutes, end of the day, summary and a sort of preview of what will be going on tomorrow. So you guys can move over to that via the schedule button in the top left of your screen. Otherwise, I will be ending the session and moving over there. So thank you all, and thank you again to the speakers. That was all fantastic.